Synthetic technologies allow scientists to venture into uncharted waters, asking unique research questions and finding previously unattainable solutions to some of life's biggest mysteries. From gene editing to protein engineering, synthesized DNA libraries enable researchers to grasp once unreachable high-throughput screening applications and dismantle barriers between experimental ideation and execution. In this podcast series, Synthetic Screens, Genes, and Novo Proteins, the scientists' creative services team talks to experts about their experiences implementing Twist Bioscience's synthesized long double-stranded gene pools, called Multiplexed Gene Fragments, for high-throughput screening. In this episode, Deanna MacNeil from The Scientist spoke with Pierce Ogden, co-founder and chief scientific officer of Manifold Biotechnologies, about the power of pairing large gene fragment pools with AI-designed libraries for better therapeutic discovery and delivery.
Hi, Pierce. Thank you for joining me today. I'm excited to learn more about your work with AI-designed binding libraries. Could you tell me how you became interested in synthetic biology and what inspired Manifold Bio's protein therapeutic delivery strategy?
I got into the field of synthetic biology and protein engineering about 10 years ago or so in 2014. It was a really exciting time in synthetic biology. There was a synergy of machine learning and what we now call AI starting to come online, but also a maturation of the methods of DNA synthesis and sequencing that really allowed us to start exploring very large spaces of genes and proteins and generate thousands to millions of variants of these molecules, but generate very specific versions of them and then assay them across a variety of ways and understand their function. It was around the time Twist was started. It was around the time that oligo pool synthesis was getting booted up. I was really drawn to that work. I was initially working in a space called AAV gene therapy and specifically focused on capsid engineering that utilized these technologies that were coming online to assess a very, very large number of mutations to the AAV capsid and understand how that affected the tropism or where the virus would go in the body. We want to take AAV and make it deliver genetic cargo to a specific tissue where it could then cure a disease. But it's quite hard to make the AAV specifically go to one tissue versus the other, like the brain or the muscle. We know that this delivery is dictated by the sequence of the capsid. And so if we mutate that sequence, we can change that delivery. DNA synthesis plus sequencing allowed us to make hundreds of thousands of variants of this AAV capsid and then measure where exactly they went. Now at Manifold Bio, we're applying a similar technology to the concept of protein therapeutic delivery.
What are some challenges you face developing and synthesizing binding proteins with therapeutic potential, like antibodies? And how do you use AI models to address those challenges?
Antibodies are one of the largest modality classes for therapeutics, but there's challenges in delivering them to specific locations, and they very often can have toxic liabilities if they get into the wrong tissue and bind their target in that healthy tissue as opposed to where you want them to be, like in a cancer tumor or in the brain. And so what Manifold's doing is working on technologies that allow us to track and measure hundreds to thousands of antibodies and other antibody-like proteins in vivo, so in animals, and understand where specifically they go, and then use that data we collect in vivo to train models and engineer those molecules further through iterative processes to hone their distribution in the body. So if we want the antibodies to go to the brain, we can make measurements of hundreds to thousands of different antibodies or binding moieties like antibodies, see which ones get into the brain and which ones don't, then take that information, train an AI model on that information, and then mutate those molecules further to try to improve that distribution and push more molecule into the brain versus the peripheral compartment.
It seems like AI tools are evolving almost exponentially in life science research. Are there any bottlenecks in establishing AI-based workflows? And if so, how do you overcome them for large-scale experiments?
When we think of the bottlenecks in the space of protein engineering specifically, with AI and machine learning, these models can handle massive amounts of data. And the models now are actually fairly well established. There's a lot of methods in terms of large language models and structural models that we can use almost out the box. The challenge is really data generation and data measurement. A lot of the generation challenge comes from synthesis of these proteins. And of course, how we synthesize proteins is through DNA. But the challenge there is that DNA synthesis is very, very limited in the length with which you can synthesize in one run. Very often, we're working with under 200 base pairs or so of synthesis length. Twist has pushed the limits of that. And that's what really excites us about the Multiplexed Gene Fragments product, is that that's starting to push the length of synthesis out to 400 or 500 base pairs. Ultimately, what that means is we can directly synthesize a much larger protein, and we can do it in these pools. So we can synthesize hundreds of thousands to millions of variants of these proteins in a single run. That power affords us the ability to make larger proteins, which are more diverse, and thus potentially bind a variety of other target proteins in cells with higher affinity or higher selectivity. And so I think the power of that technology is really in the ability to make larger, more diverse molecules to assess. This technology allows us to push the limits of what we can synthesize, and it actually still fits within the limits of what we can sequence as well. So there's a nice synergy there.
Could you tell me a bit more about how you're using multiplex gene fragments to discover and develop therapeutic protein binders? Why is it important to incorporate diverse sequences into AI design libraries? And how do longer gene fragments make this possible?
We're using this technology to make these custom binder libraries that have a very, very high amount of diversity across the entire sequence. They're AI designed libraries that have target specificities across a variety of targets that we believe are interesting for targeting an antibody to a specific tissue. But the challenge is we don't have a lot of knowledge a priori about which of these targets work. And ultimately, a lot of these AI binders fail as well because they're designed to bind the target. But what we're really trying to do is deliver them and get them to a specific tissue in vivo. So we need to test a lot of these binders to find the needles in a haystack that actually do the drug job, which is deliver the antibody to a specific tissue. The multiplex gene fragments allow us to test hundreds of thousands of these binders to hundreds of targets to figure out which ones actually get to the specific tissue. And that can be a very, very small fraction of the overall molecules tested. Out of 100,000, it might be somewhere between 1 and 10. Historically, we had used the Twist oligo pool product, which had a limit of, I think, about 300 base pairs. So that means we can synthesize a 100 amino acid protein using that product. And that's an extremely powerful product too, by the way. We've ordered tens of millions of base pairs of DNA using that product, and the outputs there are very good as well. But there are some proteins that if you could design a longer binder, could have a little more diversity in its binding interface. You can think of it like Legos. You'd have a square Lego versus a rectangle Lego. It can make a tighter lock with another Lego, right? The larger product allows us to design these longer binders, which then have a larger binding interface and also contain more diversity across that larger binding interface. And that then allows us to discover more diverse binders to more of these targets of interest. We've seen that the hit rate of the binders that are longer is actually a little bit higher. And I think that's because the models that we use, when they are capable to generate something longer, they can leverage that sort of larger Lego piece to make more binding contact interactions. It just strictly improves the process of the binders we can generate because they can be a little bit bigger. The longer the binder, the more degrees of freedom the machine learning tool can leverage to make an interesting binder. If we restrict the binders to really, really small peptides, which are, let's say, 10 to 20 amino acids, they can only do so much. They can only bind so tightly because they just don't have a very large protein interface. The Lego piece is too small, if you will, to make that many contacts. But as we make the binders longer and longer, we can really start to increase the area with which they bind to their target of interest. And so that in turn increases the success rate and the quality of the binders that come out of that experiment.
In your experience, how have technologies such as multiplexed gene fragments changed the synthetic biology research space?
We can look back over time to how people have done this sort of exploration of protein and sequence before. Directed evolution, I think, is really the start of all this work. That was basically taking a gene of interest, let's say a binder, and doing some form of randomization of the sequence using an error-prone PCR to just make random mutations to the DNA and thus randomly modify the downstream protein. And you can take that large library you've made and apply the binders to the target of interest and wash away the ones that don't bind and recover the ones that do. But the challenge there was the learning was minimal because all you had at the end were a couple of sequences that did the thing that you wanted them to do. But since sequence space is so large, the possible complexity of molecules is effectively infinite. So if you just have one or two examples of something that works, it's hard to really learn the rules and parameters with which to make a molecule. These technologies allow us to explore sequence space in a much more targeted and deterministic fashion. When we make these libraries, we have the ability to incorporate sequence diversity exactly at specific positions. So we can start to build these really comprehensive maps of how the sequences function. And then that feeds back into the machine learning models because now you have this very targeted data that allows you to fit a model and then iterate and make tighter and tighter binding Lego blocks that fit into their pockets even better. The large goal is a deeper mechanistic understanding of the biology that leads to something that seems really simple, like binding. Binding is arguably one of the simpler properties that a protein has. But even though binding is seemingly so simplistic, it's still incredibly hard for us to model and understand because of the overall complexity of the amount of different ways that you can make a binder, of the complexity of sequence space, the ability for us to generate this data at high throughput in a very deterministic fashion allows us to characterize this landscape and sequence space and then leverage that information to understand it. And hopefully, as we generate more and more of this information, we can start to actually build things like the protein-protein interaction map that have a holistic understanding of the mechanisms of how proteins bind, and then we can leverage that to make new medicines for patients.
I have one final question for you. If you could synthesize DNA of any length and any complexity, what would you build?
I'm obviously a very biased individual because I've been working on delivery of cargoes for my entire career. The reason that I've done that is I think that there's just so much improvements to be made. Folks have been working in these spaces for a long time, but they're just fundamentally restricted by the fact that delivery is really hard to measure in vitro. So you really need these in vivo models to enable it. And in vivo models before synthesis and sequencing were very, very slow. So you had to just make, one delivery vehicle, whether it was a virus or a lipid nanoparticle or some sort of targeting domain for an antibody, and assess it at once. But I think that if I had any length molecule that I could generate, I would probably look to make something that looks like a synthetic delivery, let's say nanobot, where maybe you would take some of the powers of something like a protein cage. These things are often thought of as like VLPs, virus-like particles, etc., that then could package significantly larger cargoes. So those could be antibodies, they could be combinations of antibodies, they could be oligos or DNA. Then I would try to enable that to get very, very specifically into not even tissues, but certain cell types, so that you could deliver that cargo in a highly specific and efficacious manner. If you want to target a certain cancer cell population that has a specific pattern of receptors on its cell surface, this would allow you to do something like that. Or if you want to target a very, very specific cell in the brain, you could then do that as well. I think that ultimately is my dream and vision, and I'm excited for these technologies to continue to improve. But there are a lot of interesting questions, other applications that I think folks could be interested in that are unlocked by these technologies as well.
It sounds like there are a lot of interesting avenues ahead for these technologies. Thanks so much for sharing your time and expertise with me today, Pierce.
This has been a fun conversation, and I'm excited to talk about multiplexed gene synthesis, something I've been working on for basically my entire professional career. And it's something I hope that more and more people get exposed to, because I really think it stands to fundamentally shift and revolutionize the way we do biotechnology, the way we develop therapeutics. So yeah, it's been a lot of fun, and thanks for having me on.
Thank you for listening to the final episode of Synthetic Screens, Genes, and De Novo Proteins. And thanks again to Pierce Ogden, co-founder and chief scientific officer of Manifold Biotechnologies. To keep up to date with this podcast, follow The Scientist on Facebook and Twitter, and subscribe to The Scientist Lab Talk wherever you get your podcasts.